Thank you, everyone. My name is César Marín. Here you see my CV a little bit. I did a couple of postdocs over the last few years, and now I am starting a permanent position here in Chile in Santo Tomás University. I have a PhD in ecology and evolution, and when Daniel asked me to make this presentation, to be honest, most of my research has been based on temperate forests. I have some six or seven articles on tropical ecosystems and mainly on the mycorrhizal and the fungal side of the symbiosis. But I am also the leader of the South American Mycorrhizal Research Network. We have now like 5 years and 275 members. So I will talk a little bit about that. The network is integrated to several other networks like in FAO or in the International Mycorrhiza Society, on Soilborne also. We collaborate with several global scale projects like the Global Soil Microbiome and the Fund Leaf regarding the phylosphere. We have a YouTube channel which is very active, like one interview a month for mycorrhizal papers. And like 3 years ago, we edited the first book on mycorrhizal fungi in South America. This book has like 17 chapters, very, very interesting chapters. And we have organized a couple of symposiums, one in Valdivia, the city where I did my PhD, and the second in Bariloche in Argentina, very nice keynote speakers. You are all invited to attend the next one, which will be in Leticia in the Amazon in Colombia in 2023, August of 2023. And why they need to do like specifically a South American thing? Because when we compare like the amount of research produced here in South America, for example, to Europe, is way less, despite having way more land area and way more like ecosystem diversity. And also the focus of each continent has been quite different. Here in South America, we mostly focus still in community structure of mycorrhizal fungi, while in Europe, the focus is more on anthropogenic effects. And the graph that I show below is a screening of the fungal traits database. This is a really recent database which classifies several thousand genera of fungi into different functional guilds. And if you see in the graph that arbuscular mycorrhizae and ectomycorrhizae have super low numbers compared to in South America compared to the other continents, but this is just a lack of research. This doesn't say that we have less diversity. It's just that the amount of inputs on this database from South America is super low. So we are still in a phase of building these databases and knowing what we have. My master student in thesis, Javiera, she did this really nice compilation about different mycorrhizal types, different ecosystem functions provided by mycorrhizae. And in the blue dots are represented the studies where both things are studied at the same time, biodiversity and functions, which is quite good. It's at 24.21% of the sites are studied at the same time. This is quite significant compared, for example, to global studies on soil ecology, where this percentage is like 0.03%. And anyway, you see that the focus has been mostly in arbuscular mycorrhizae, almost 1,000 sites. Then something more in ectomycorrhizae, and the main function study is plant growth, basically a little bit soil respiration, but the other functions provided by mycorrhizae are quite low. So to counteract this lack of information, and for the interest of this group, you see that, of course, a lot of blank spaces in the space in the Amazon, and also in the Cerrado in Brazil, very few studies. So we need to work way more on those places. So to counteract this, we did like a first workshop to build this database, both with literature, with Web of Science articles, but also with gray literature like Spanish thesis and Portuguese thesis or publications. So we are in this phase of building this database with all the steps that this requires. And this is like a first example of the kind of things that we want to both look in the literature, but also sample and measure. So you see that we have basic information, but also a lot of mycorrhizal information from the mycorrhizal type, status, a criterion for diagnosis of arbuscular mycorrhizae, amount of arbuscules, and then of course some plant information and some soil and climate information. So I think that will be my presentation. Thank you.